Okay, we are about 50 meters from bottom, and this is NOAA Ocean Exploration EX 2107 Dive 11, which we're calling Key West Scarp. We're about 40 miles due south of Key West and heading for a bottom depth of about 1,160 meters, and where there's a small wall of about 50-meter rise, and we'll be ascending that wall, looking for what's living on the wall, and then doing a transect along the top edge of the wall. I am Alan Collins with NOAA Fisheries and the Smithsonian National Museum of Natural History, and I am with Stephanie Farrington.
Hello. What do you got, Alan? What is that?
It looks like a shrimp. It's a lazy shrimp.
Well, you don't really have to work that hard out here because the currents just take you. Super chill life. I don't know if anyone has ever been drift diving, but it's some of the greatest diving ever. So.
There we are. Bottom
right.
is in sight.
It's 100% sediment.
Yep. A few burrowed holes, so it's bioturbated sediment.
There's something white there in the middle.
As soon as we get situated, we'll take our second water sample for eDNA, and then we will proceed to move towards the base of this
Hopeful
wall.
feature. Yeah.
Yes, hopefully it's there.
2 days ago we got confused, but yeah, hopefully we have one today.
Well, we were confused because things had changed.
Yeah. Very odd.
Shows how dynamic the seafloor can be.
These little long, skinny things are probably detrital seagrasses from the shallows. Not uncommon when you're this close to the Keys. They die and float out here. Become part of the carbon cycle.
Yeah. So I think we're still going to do our white balance.
I don't know. Did he do his white balance yet? He didn't. Yes.
Maybe, but it can be after the white balance and stuff.
Yeah, we're going to settle in, do our camera balancing, white balancing, and then take a water sample. So it'll be a few minutes. Copy.
Sounds great. Thank you.
Correct.
Correct.
It's fairly flat. I only see very little bioturbated sediment. Yesterday it was a lot heavier. Maybe in the distance there's a little bit.
Yeah, it's here and there, but
Yeah,
you're right.
Not nearly as highly concentrated as yesterday.
Nor are we seeing much in the way of ripples.
Yep, which means the sand at the bottom, or the water movement near the bottom is pretty low. Low current. Which you can see in the water column, kind of passing by a little bit.
Slowly passing.
A little red shrimp or something.
Yep.
Yeah, left to right, we're facing northwest. So that means the current's coming from the southwest, roughly.
Mm-hmm. And much more gentle down here, apparently, than, than up top.
Yeah, but you're not current at the surface. So if you've never tuned into our video feeds before, this is something we do every dive. There's this special color card that D2 had made special, or GFOE had made special for color correcting our cameras. So you'll see it's attached to our arm. It has all the different colors on it, and blacks and whites and shades in between. So we use that to check the white balancing and the color temperature of the cameras, and you'll see it zoom in, and our main camera, which is feed 2, and then after a while the card will turn facing up, and that's actually color correcting our sampling camera, which I don't think is being fed out right now. So sometimes it'll get fed out on on feed 3.
There are a lot of cameras
Yes.
on D2.
Absolutely. And not even including the ones that are on Sirius,
Yeah.
just on feed 2. Oh, feed 3. Okay. Or not anymore. You may have missed it.
That's okay.
Yeah. So it'll just be maybe another 5 or so minutes. Maybe a little longer. Till we get situated, and then we'll start the dive.
Every Wednesday is Worm Wednesday. Maybe we'll see some worms today. Not all of us love worms as much.
No. They're perfectly fine existing. I just don't need to look at them. That's all.
Well, we'll see what we find.
Mm-hmm. The ones that look like little mice are not that bad.
Mm.
I don't know what they're called, but they look furry almost. Yeah. And the ones in the water column don't bother me that much either. I don't know what it is about. It's the way they move.
It's the way they move with the lots of legs.
Yeah, it gives me the willies.
Yeah. You feel that way about centipedes?
No, not really.
That's all right.
Roaches, though.
Yeah.
And I lived in Florida for a long time. They fly very badly.
Yes, they do. Yes.
Horrifying. Like a monster.
like cicadas fly kind of poorly.
They do, but I don't know why cicadas don't bother me, but roaches do. I guess I never had cicadas in my apartment.
Mm. It was pretty fun to see the big brood 10 cicadas this year in the DC area.
Mm-hmm. I like the noise they make. Yeah.
Yeah.
It's really cool. Oh, I see a red crab.
Chase. Is the chase on?
For Chase
on?
Yeah.
Yeah. I shouldn't say it out loud, but seeing Bathynomus on the soft sediments the other day was pretty darn cool. I wouldn't mind seeing another.
Yeah, they're not uncommon out. I mean, they're no more, they're not crazy common either, but they're not uncommon out here on the sand.
For me, that was that's one of the visual highlights of this expedition for me so far.
Check off your list. I like the mini peta we saw yesterday, right? The squid? Did I get the name right?
Yeah, the Magnapinna.
Magnapinna, thank you.
The big fin squid. Yeah, that was pretty spectacular. We've actually had some nice cephalopod sightings throughout. All the patrols of the Elix Squid.
Yeah, that was good.
That was pretty nice.
Down here in the Keys, they're common down here, migrating or something.
Mm-hmm.
They're probably the other most common cephalopod we saw for sure. But they're pretty common just among the other animals too. That day.
And the flapjack devilfish. Pistoteuthis. But then also seeing those egg cases from that same group yesterday laid on Gorgonian branches. That was
neat.
That was pretty cool. I wonder how many people are going to be watching, searching for Magnapinna off in the distance, because that was so so extraordinary to see.
Yeah, that was awesome. That's definitely a highlight.
Yeah.
Yeah, that big black eye. You can see the nostril.
Mm-hmm. I think we are cut off from the chat room, so the chat room may actually, people may already be reporting exactly what this is.
Thanks, Roland. Yep.
Great. No, it's definitely
So there's two kinds of egg laying. There's oviviparous and then there's oviviviparous. And oviviparous means that they have eggs that grow internally and they lay eggs, like a chicken. Or oviviviparous, where they actually lay eggs internally and then those eggs hatch internally. And that happens a lot in sharks. So, one of the two. There he is up there. Or she, I guess.
So if you got cut off the feed, we did see an opistoteuthis on the bottom. That's the flapjack devilfish, an octopod, quite cute. And then we just were looking at an incredibly, well, a fish that was very unusual to us.
That was likely pregnant. There's something white ahead and something moving, maybe a squid.
Yeah.
Elix squid.
I think that is Elix, yeah. So, Elix is a squid that aggregates in this region during this time of year for mating. And they'll spawn, actually, excuse me.
And that white thing. Maybe just be human debris. Yeah, more plastic. Maybe a shrimp in the midwater.
Coming at us pretty fast.
Ooh.
Whoa!
Bye.
we thought it was fast before.
It's a flat sandy bottom. We're approaching our waypoint, which we should see about a 50-meter rise when we get there. This is what most of the bottom of the ocean looks like.
As in covered with sediments?
Mm-hmm. Sandy beach. I got something at 9, the distance. Yeah. Probably just sargassum pile from this distance is what I'm seeing.
Mm-hmm.
Yeah. Okay. Thanks.
This may be a wall we actually are able to climb to the top of.
Yeah. We never seem to make it. I think there might be some human debris here at 6, 7, 8. Like some line or something. Yeah, line.
Looks like line.
A little bit of plastic, maybe. Human debris off the Keys is not uncommon. There's a huge tourist industry in this area, so you end up getting stuff floating down from the surface.
Mm-hmm.
We're only, 30 miles away or so, 40 miles from Key West or so.
But even when we've been quite far out, we've also encountered human debris. I won't forget the soccer ball at the bottom of that sinkhole.
I know, that was crazy.
Hundreds of miles from shore.
Yep.
Anytime too soon.
Yeah.
Just a nice reminder that our activities matter.
That's right. There's maybe some hard substrate, or looks like you have a little shrimp in the foreground. It's kind of blurry. Oh, it might be squid ink, actually.
I think that is, yeah. It's really very yellow.
This is a little divot here with some sargassum.
Who's that on the ground?
Oh, we got something in the 9, 10 o'clock in the distance. Shark-looking thing. Yeah.
Yep.
Yep.
It's a shark, all right. Let's see. I wonder what kind of shark.
Dogfish, or it's a pretty flat head, really reflective eye. So on the back of the eye, they have something called tapetum lucidum, which actually reflects light, so they can see better in the dark.
I'm wondering if it's a gulper shark.
It could be. Oh yeah, definitely. Centrophorus.
Centrophorus granulosa, maybe. It's coming from someone who doesn't know that much about fishes. An invertebrate zoologist.
Yeah. Another possible Pleodicus behind it, the royal red.
Don't often see sharks just motionless like this on the bottom, says Mike Vecchione.
There you go.
Looking for spines on the leading edge of the dorsal fins. I don't see spines, do you?
There might be one right there.
Oh, yeah. Yes, there is a spine there.
So that is one of its identifying characteristics. We can get it down to species, maybe?
That was...
You can see his gills moving. That's neat.
Centrophorus have spines, so we may have actually got it.
Nice.
Really nice. Look at his eyes.
So I was always taught as a kid that if sharks stop swimming, they die. And I don't think that that's true.
Well, it depends on the shark, right?
Like, obviously it's not true for this guy. He's
Yeah.
doing just fine.
Yep. Kevin Rademacher says that there's actually there's a lot of undescribed species in this genus, and so it's probably premature to identify it to the species name.
We can go to genus level then.
Yeah.
How about that?
But, yeah, there are definitely some sharks that will die if they stop swimming, but I think a lot of the benthic ones, obviously this guy.
A female? There's no claspers?
Mm.
Yeah. So likely a female.
Up to 7 undescribed species, possibly.
I've been misgendering pretty much everything out here, so I apologize to the wider marine world.
Well, that was nice. That was nice footage of that shark. Seen some great vertebrates so far. I want to know what that one fish was that we saw in the water column just above the bottom.
Oh, yeah, I don't think anybody... That was when we were
The
cut
one that
off.
looked all pregnant.
Yeah. Just setting that out.
Somebody else on the bottom there.
Yep.
Is that a Elix again?
Could be. Up on his elbows. Another little shrimp. 7. Yes, definitely a squid.
Yep, that's Elix. Oh, and there it is, sitting in its... characteristic, and it's perched up like that to keep its funnel off the bottom so that it can...
Not suck up a whole mouthful of sand. Note from landside, if you are on the conference call, please mute your phone if you're not talking. Thank you. So you can see the chromatophores, the little brown circles. That's where they change color on their skin.
Mm-hmm. And once again, these are aggregating in these waters for spawning. It's about spawning season for them.
Mm-hmm.
If you look at this,
did you see him change color?
Yeah.
That was so cool.
That was so
Sorry,
cool.
go ahead.
That's all right. No,
I interrupted you. I didn't mean to
no
do that.
worries. And there's
one
another.
above?
Yeah, so we'll probably wind up seeing quite a lot of these Elix. The last time we encountered them, they were the most abundant organism.
Can you spin 3 o'clock? Yep, right near the squid. Yep, can you go in a little bit? I just saw something move and a little bit out. So, yeah, a little bit out. Okay, on the bottom there. Okay, let's see, 5 o'clock of laser. So just spin a little. Yep, there we go. 3 o'clock of laser. So just spin a little to the right, and then right above laser, right there. Okay, now to the left. Yep, keep going a little bit left. That. What is that? Oh, it's a scallop. It's a scallop.
Oh, so it swam.
Yeah.
Nice. A bivalve, a scallop.
Pectinidae. Nice. Thank you. There's a couple of them, actually.
I know some have eyes, but I'm not sure
Yeah,
if
they
all
do.
pectins have
They
eyes.
have like a lot of eyes.
Yeah.
You got something in the water
Yeah, can
column?
we check what's in the water column?
There goes the scalp speeding off.
Oh, there you are, Jelly.
Oh, he's tiny.
He is small. I think that's a Ropalinamatid again.
You're going to have to spell that one for
I
me.
I will. Oh, yeah. You can see it's got
cool.
8 gonads. It's flowing down slowly.
What's the blue? Is it a reflection
I
of?
think it's just, iridescent reflection. Thank you for getting that. That's great.
Tiny. Another cutthroat, maybe?
Should be able to refine that ID later.
Looks like something sped off to the left. Cloud of dust. Oh, yeah. There's something. Keep going left. You can see it in Sirius's view. Maybe a fish or something.
Another shark, maybe?
Oh. How are we not seeing that yet? It looks like a cusk eel. Oh my goodness, it's right there.
Is. Wow. Okay, let's watch this a bit.
Yeah, turn sideways, man. You can do it. Come on. Pretty hefty fish.
I'd love to see its head again.
Mm-hmm. It's going to settle down on the bottom, maybe?
It's hiding from the lights, I guess.
Really large head.
It does have a really large head.
White eye. Maybe half a meter long.
He's just chunky.
He is pretty chunky. He is dead set determined to not look this way. Deep sea troll.
I will not be identified.
You bugger. Where are you going?
Yeah. That's.
Come on, keep turning. Oh.
Well, ooh, is it going to turn for us now?
A little bit. Keep going. Look at his mouth.
Yeah.
Thanks, man.
Oh, he's behaving.
Looks like you got a mouthful of sand. That's a clouded eye.
I think this will be good enough for an identification. What do you think?
Yeah, I hope so. He's very hefty.
Yeah, I think that's great. That's going to be great. Thank you. Just making note that there's no barbel, apparent absence of pelvic fins. So,
Hey, you're back on the phone. Good to hear you.
Hi.
Hey, Scott. Welcome.
Welcome to you.
We've been missing the phone chatter.
Yeah, actually, it was kind of fun because Mike called in to me to give information. Yeah, it's good. You've been doing a fine job in our absence.
Well, to talk about what we don't quite recognize, that's not too hard.
Yeah.
And we're seeing really cool things.
Well, that's good. I mean, just saying out loud what most people watching are experiencing, right?
Exactly.
What is that?
Well, there's something on the bottom there.
Divot
Is
or
that
something? Cloud of sand in a water column. Something white far away. You got a Medusa at 3.
Oh, nice. I think that's that same one. Maybe.
Yeah.
Any chance we can grab this Medusa? Okay. Yeah, as a sample, that's what I was wondering about. That might be a little late. I think he's going to go by.
Ooh, bye.
He is pretty cool. I think it looks Cresota-like. If you happen to notice one not far out, and it's not too high, might be nice to... Thank you, pilot.
What's this white thing at 4? Went past it. There might be a worm there, Alan. At 4 o'clock. Yeah, down. Yeah, towards the corner of the screen.
Well, it is Worm Wednesday.
There's something smushy. Another red shrimp. Yeah, it's right at 2 o'clock. A laser, right in front of that shrimp. Yeah. Oh no, another scallop. Yeah. Okay, thank you. Pectinity.
Yeah. I'll get a little close up on it. That's great.
So this is a bivalve.
Mm-hmm.
Probably not the scallops you're eating at the restaurants. Relative of those, though.
Yeah, I can't help but think it tastes good, though.
More squid up in the background.
It goes another jelly, Medusa. So we're making our way to the wall we expect to investigate.
Mm-hmm. It's at 1,110 meters. We really haven't changed depth much. We're still flowing along on the flat area. Doing a little bit of a pilot change. A little bit of... Go ahead.
No.
Go ahead. Do you see something?
No.
Okay.
Something pelagic floating by. Nothing worth the... Couldn't see it that well.
Something schmutzy.
Yeah.
There's something at 6, now 5 o'clock on the sediment. And then a shrimp at 9. Some detritus orgasm at 6. Little tuft of algae that's died and sunk. And then that red shrimp, which I'm guessing is Pleodicus, but I might be totally wrong. Yep, at 3.
At 3 o'clock, yeah.
Mm-hmm.
So we're out here. Mike Vecchione suggests that we look for big gelatinous egg masses.
Sounds good.
Which would be a little higher up in the water column. And then these egg masses actually drift in the Gulf Stream, and that's where they hatch from the squid, the Elix squid that we've been looking at. Let's take a look.
That X chromosome thing.
No, I think this is different.
Is that a spider?
That's exactly what I was thinking. It looks like some spiders hold themselves.
Amphipod or something.
Isopod.
This is an
Isopod.
isopod, one of the water walkers. Nice. I
think you can see the pleopod swim rest at the first, so it's pointing downwards.
And the segments in the legs. Ooh.
Now here it comes.
Oh, what's behind him? Some kind of eel.
I think those are isopods or munopsids. M-U-double-N.
Menu.
Curious if we ever collected one of those on this Okeanos.
The sample collection list is on the server. Sure, we could find it. something pink. There you go.
Ooh.
Isopod, amphipod.
I think that's another isopod, yeah. Oh, it's so neat. Yeah. I think, Karen Osborne, our colleague at the Smithsonian, has done some work on these munopsids. But mostly out in California.
Oh, yeah.
So
There's a sea star dead center.
let's take a look at this star.
There's actually a footprint right here, too. Yeah, there's one.
Nice.
Yep, there you go. At noon.
And I can confirm that we do have Chris Ma in the chat room, so he may tell us more about this
Maybe
star in the
Lydia
sand.
or something?
It's funny, it's definitely down in there.
Mm-hmm. I think it's a goniasterid. Some type.
That's a nice close-up view. That's Zoroaster fulgens.
Oh my goodness. It's what now?
Zorro. Z-O-R-O. Zoroaster fulgens. We've seen that, I think, once before. You shall not pass.
Zoroaster. Is he named after, like, the character Zorro?
I'm not sure if it's that or Zoroastrian, or both. Chris could tell us.
Yeah. Maybe a rock up here? Another rat tail.
So our first real structures.
Mm-hmm.
On this rock, we'll almost certainly see some small encrusting demo sponges.
This looks more like a broken old coral rock.
It does.
Right. There's human debris. There's a crab at 9 underneath the rock. There's some human debris towards the right. Maybe a couple of sponges in the middle. No, it's okay.
And keep moving to the wall.
Yep. Little fishy, rat tail fish.
Yeah.
He looks similar to Zumia, but he's got very long, tendrily fins. Very ripply tail. Has a parasite on its back.
Yeah, we've seen quite a lot of parasites on the fish. I wonder what the rate
there's some worm tubes or something at 9. Sticking out of the bottom. Is that what I'm seeing? Worm tubes? Something like that.
I think that must be worm tubes.
Mm-hmm. A little cluster of them.
Zoroaster literally translates to whose camels are old.
What?
And it alludes to its very ancient appearance.
There's a bivalve shell?
Chris Ma also clarifies that Zorro is with 2 R's.
Oh, okay.
And that the name is from 1899, so it actually predates.
Zorro.
Yes.
We have a yellow sponge on that rock. At 3. Yeah, the one on the rock on the right. There's a yellow sponge. Yeah, that guy right there. Hexactinellid, maybe?
I think maybe.
Hard to tell. It's very fuzzy looking.
Hexacs are usually pretty clean. Mm-hmm. Oh, it's got zoanthids in it. Yeah, and I think it is a glass sponge. Sort of farreid, urated. It's all on a big clay called Sceptulophora. And lots of zoanthids in there.
Yeah. I don't know. I don't think we've ever figured out why some of them are infested and some of them aren't.
Yeah.
Sometimes.
Yeah, we actually had a request for zoanthids on different glass sponges. Is this a...
Want to collect it?
Yeah, can we collect this?
There's also a small gorgonian on the other rock.
It'd be nice to... Evidently these zoantharians that are on make associations with the sponges.
Yeah, ideally, yes, if you can. It's going to be very fragile. They're going to have to back the ship up.
Oh.
How long does that take? Is it like a It's up to you, Alan.
Yeah, let's get it. I haven't gotten anything else. And since we had a good justification, special request for this. So the primary sample is going to be, I think at the family.
Feriidae?
Yeah, let's stick with that. But obviously, that's just a field ID. And nice. Gonna line this one up.
No, like crab in there.
Squids have been hiding behind D2. And it's likely to be quite fragile. If it's small enough, you could use the suction sampler, like bump it and break it. But
Alan, can I request we get one more zoom on it before collecting from this angle?
Could you say one more time?
He wants a zoom.
Yeah, okay.
I asked, could we get one more zoom on it prior to collection from this new angle?
Great.
Can you guys snap zoom the sponge first? Before we start.
Scott, do you think this is something you've had picked up before?
Oh, no. This is great. I think this is a really good collection target for all the reasons you pointed out. Interesting looking sponge, the zoanthids on it. We've had special requests for these associated symbiotic zoanthids. So, no, it's all good. I just want to be sure we see it really nicely because, as Stephanie said, it's going to be fragile, so it's going to be quite mangled once we get it.
Right. Great. Thanks, Scott.
Awesome, guys. Yeah.
This
is going to be super fragile, so your best bet's probably to take your clippers and go right all the way to the base, or to pick up their suction hose and just use the edge to scrape it off with a very gentle suction.
So that's a really nice view, and one of the things I wanted to see also was the zoanthid polyps themselves, because we know that once we present those, really contract. So that's a really nice image.
Right.
There's also a bamboo coral on the rock behind it, Scott. I don't think you heard me. There's a bamboo coral on the rock behind it, too. Oh, yeah, for sure.
So, Chris Diaz says see these little white puff balls?
That's good, thank you.
Those could be Leucopsacus.
Leucopsacus.
Yeah. Just below the octocoral.
Mm-hmm. I don't see any rings on that.
No nodes?
Mm-mm. They look more plumarella-ish, primnoidy-ish.
I was typing in the chat. I was typing in the annotations. I wasn't paying enough attention, but as we were going in, I agreed with your first ID that was a bamboo coral, but I'll have to go back and look at it more closely.
I'll remember to take another look at that.
Says this is probably less than 10 centimeters, yellow, with zoanthids. More squid ink in the water there. What does the crab have? Is that half a fish? Oh, it's squid. It's like going to the grocery store. Oh my goodness. I'm going to take my squid and go home, thank you.
Now we'll have to see if we get anything else on this suction.
It looked like there was a...
There was a lot right around there.
And right next to the sponge was a barnacle,
Yeah.
a gooseneck.
So just hoover around.
Stop it. One sample. Oh my goodness. We got some serious deep sea predation. Looks like the fish to the left has also got himself something. Yeah, this should just pop right off. It should be super easy. Just a little bit of a tap and at the base, and it should just pop off the rock and go right in the tube.
You see that little white sponge in the background? Maybe we'll zoom on that and that octocoral, the non-bamboo.
Yeah, I don't know. I don't know what it is.
Yeah.
The zoanthers are still out. Once we collect it, they'll probably pop in.
Yeah.
They just kind of ball up into little nubs.
So if there are dead squid in the area, that would, Mike Vecchione suggests that means there's spawning going on.
Oh, okay.
So.
Yeah, that was a good catch by that crab that got stolen by that fish.
More good carbon transfer.
There we go. You can see the suction moving in the hose.
Got a geological sample.
I think the gooseneck came with it.
Oh, good.
Yeah, you can see it's pretty loose. So once it just breaks free, it should just get right in. There you go.
Oh, how wonderful.
They're really fine. Oh, that looks like a sea star behind it.
And there it is. It's in. Nice. Nice work.
Yeah.
It's
Number
canister one.
Mm-hmm.
Great. Well, that was exciting. So we're here, keeping track of all the organisms or all the samples we make. Great, great job. So I think there might be a star tucked in that little crevice there, and right in front of us.
Yeah, let's get a zoom before we speed off. Oh,
Science, is it those white things that you think is a star?
yeah, it's a squat lobster.
Oh, is it a squat lobster?
Squat lobster from
Yeah.
my point
Yeah.
of view.
Yeah, I can see it from above you, from the camera.
Good.
Over here.
You guys have your years of experience.
Is that a fish on the left? Or a squid arm?
Squids, they just seem so curious.
There's a cutthroat eel, I think, lying on the ground at 8 o'clock in the hole. He was wiggling around a lot earlier.
Thank you very much. Great collect. Yeah, it would be great to relook at the coral that was just behind that sponge.
That guy, yeah. There might be a ring at the base of that one branch, but it's tough to tell.
Yeah, it looked.
No, Stephanie, I think you are right. I think I see on the polyps there's the appearance of scales. You can see these lines running down the length of it. And the skeleton itself, that color, it's a primnoid. It's not a plumarella, but I think it is a primnoid. It's a good call on that.
Primnoidy. Yeah, thank you.
Yep, thank you.
We have some of those blue sponges we collected the other day too, the encrusters.
I think, yeah, hopefully we'll see a little more of those white, small white, like a little, like an
Sponge?
egg-shaped sponge,
Yeah.
really tiny.
These like rocks have all these holes in it. They look like typical like coral reef rock, right? It's old coral reef rock.
Mm-hmm. Who's that?
I'm not sure. What's that? Yeah, we're starting to see a little bit of a wall ahead of us.
Mm-hmm.
That might be human debris, I'm saying.
I think it is.
That looks like a chrysogorgid from here, at top of the
Yeah.
rock.
Some sort of a belt was on the ground.
No, it's not. It's a fish. Okay, he's going to be pretty cool.
Oh, yeah.
Yeah, you see Anthomastus too? This is
Is
some
this going to
kind
be
of...
Chonax, or maybe?
I'm not sure. He's some kind of anglerfish, I think.
Oh, okay. Great.
Hey, anyone know the name of this fish?
I don't remember it, Stephanie. That was me calling back in.
Oh, okay.
Bounce from the conference line for some reason.
He's pretty neat.
It's going to
I
be
love
this guy?
Yes, that's it.
Schaefer's anglerfish.
That's it.
Sladinia Schaefer's eye. Yeah. Maybe we'll get a little look at it. I guess you can see his barbel a little bit on top of him. Sladinia.
Yeah, his barbel's on top of his, between his eyes.
Uh-huh.
They look like they stand on little feet, little modified fins. Obviously an ambush predator.
Oh. That's an Esca, not a barbel.
Oh, what's the difference?
Kevin Rademacher corrects our terminology, which is very helpful. Those spines around its mouth.
Running up at the back of the leg, along its back. It sits perfectly still. It has a humongous mouth.
Yeah, it looks like all mouth and stomach, really. Those modify fins for holding on, that's really something.
Yeah, that's cool. It looks like there might be some corals in front of him on the rock.
Yeah, that's great. What a shot.
Yeah, it's cool. You got something in the water there, Alan.
Yeah.
Little red guy.
Oh. He's the king of the mountain. We could look at the just below the fish on the rock. I think that might be another yellow sponge there.
Yeah.
Yeah.
Do we want to predict if it has zoanthids before you zoom in?
I'd say no.
Oh, I'm going to go with yes.
I say yes.
Ooh, we can see it. This is nice to see the structure of the sponge, but you're right, there are no zoanthids here.
Man, Stephanie's on a hot streak today.
He does look like he has a commensal animal inside him, though. Is commensal, am I using that word right?
Yeah.
Yep, inside at the bottom, it looks like some critter living in there.
And it's interesting how it's the pigment is concentrated in the lower portions of the sponge.
Looks like maybe a worm. No pink.
Yep. And then the...
maybe a really small cladorizid at the bottom left of that sponge. A little spiky ball.
I think it's
a worm tube? Or what do you think?
No, this I think is a barnacle. It looks, it's like this, right?
I
Like
see.
2 hands.
Uh-huh. Thanks, that's great.
Yeah, we're good whenever you guys are. Maybe some, like a coral rock or rubble in the bottom. There's a chunk of coral below the fish at the top. Just a really quick snap zoom, just so we can quick ID it, and then we can go.
Thank you.
Yeah, that looks like hard coral to me.
Sure does.
Wow, that's neat. It's not Lophelia, I don't think. It's very Lophelia-esque.
The angles are sharper, right?
Cool.
So Lophelia-esque, but I'm really interested in the primnoid behind it, and I'll just describe it. You notice that along the branch there are several places where it looks wider. Those are worm tubes along the branch. And so there's probably polychaete worms living in there. There's a whole, at least a genus, maybe in a family that's specialized. It's not a family because they're scale worms, but they're specialized to live in these corals. And when they land on the coral, they induce the coral to deposit its sclerites in the form that makes kind of a little tunnel or a garage. Is the way I like to think of it. So the polychaete can live inside there.
Nice.
Wow.
Okay, we're good whenever you guys are.
Candidella imbricata is the name that Steve Oskovich.
For what?
For the primnoid.
Oh.
I presume.
Yeah, we see the rock ledge coming up ahead of us.
Nice that we're getting to the wall.
Sticopathes and Anthomastus.
Anthomastus, or Anthomastus type, I guess.
Here's another pharia with zoanthids. Some of this fresh exposed rock is white. The old stuff is black. And you see the layering in it.
Oh, this is nice. This is definitely harder, more consolidated than we saw on the Tortugas Valley site.
I was expecting it to be that soft mud, that consolidated mud. This is actually real rock.
Yeah.
Probably reef, old reef. Yeah, there's a fish at 9.
Take a quick look at that fish. Yes. Is that a cucumbers? I think that's a halosaur again. Yeah.
Underside.
Yeah, definitely a different view. You can see how looks like the lower jaw is so much smaller, right?
Mm-hmm. Pointed.
I wonder how often they're seen from this angle, since we've encountered them on the bottom, right on the bottom, and they're bottom feeders. Now the fish is not happy.
So we're approaching the base of the wall.
One of the red shrimp in the water column.
Mm-hmm. A mysid or something. Lots of pock marks in the wall.
Mm-hmm. I think that was a mysid.
We have a Antipatharian at 2, just off screen there. Sympharia. Oh no, sorry, it was, if you go full wide, it's up above you at 1 o'clock-ish, near the edge of the screen. Yeah. Looks like it's got a big crab on it. Squat lobster.
Sure does.
Mm-hmm. Really long legs. I also see a spiraled Stichopathes at 3 o'clock.
There's quite a lot of sediment on this wall.
What is that? Sea cucumber.
Yep, there's a sea cucumber, a swimming guy.
Where you going, bud?
Another one of those yellow Phoreas. So that's turning out to be pretty distinctive of this site.
Mm-hmm. So this is like a pink-hued, my guess is Antipatheria from this far away. It's sparsely branched, really fine pinnules on the side, with a very long-legged squat lobster in the middle. Looks like another shrimp there.
It's got a shrimp on it. Scott says it might be Paranthropes. Parantipathes. Oh, nice zoom on the shrimp.
Parantipath. So many of these black corals have pathes or antipathes associated with them.
Mm-hmm.
But, this closer look, I'm not so sure that it's that genus, and I'm not as familiar with these more shallow. I mean, I realize we're at 1,100 meters, but some of the black corals. I'm not sure if that's it. It could very well be, and one thing that's interesting is that genus is very often associated with a pair of chirostylid crabs, these little squat lobsters that are specialists on living on corals and sponges, but not like the one that I see there. That's a pretty large one for what I'm used to seeing. It's at least not the genus Uropticus. And I'm also not used to seeing shrimp on them, so that was an interesting observation.
Mm-hmm.
Here we get a nice close-up of this squat lobster with the really long legs. And
yeah, you can see the modified back leg is like hooked, right? So they hold them up hooked behind them so they can attach like that. Yep. Get a good grip.
Get a grip, Steph. Wow.
Very spiky.
Yeah, you don't want to grab that with soft tissue.
Right through your finger. He's cool. Oh, look, you can see his really fine pinchers. So his chelate arms.
Mm-hmm.
Almost like tweezers, like you reach out and just...
For snatching
yeah.
prey out of the water column. Or picking stuff off. Looks like a happy crab.
Yeah, I think so. Good place to be.
Shrimp. That's an interesting...
Little shrimp guy. Cute.
We got another associate there on the blade at, you're coming towards it. It's about 7, 8 o'clock.
Oh, yeah, small. It's hard to say. He blends in with the antipatharin really well. And cream colored.
Oh, no, it was
at the bottom.
Down below. If you go down. I think there's something on that one, too. And it's on the blade just to the right.
On the black coral.
On the black coral.
Right there, center screen. No, not noon. Yep, off screen now. It's fine. Do you want to get a zoom on it, Alan?
If we're still here, why not? So
yeah.
Straight in on that blade that's turning to the left, and then you're coming right at it. Yeah, really small orange, blends really in well.
Little shrimp.
Just a bit to the right. Is that a shrimp?
Yeah, I think so. Yeah.
Oh yeah, nice.
A little pinnate or something.
So more than one shrimp on this. There it is. Thank you very much.
Little guy.
I also saw one.
I don't know
on the
about
other
the
two.
shrimp because they're so mobile, but those parantipathes that I was referring to that we see out in the open Atlantic and the deep waters, when we find a pair of these squat lobsters on them, there's these Uropticus. It's typically a male and a female, and I think that adds to this idea that chirostylid squat lobsters, they don't swim around much. They find their coral, and then that's their home, and they hang on. Stephanie noted those back legs that are hooked and ideal for hanging onto the colony. But unlike the shrimp that can just sort of, swim around and find a nice place to settle, those crabs find a spot. So when you find a pair, they're presumably a mated pair for life, is at least the way I'm interpreting it.
That's
Nice.
cool.
There's some Stigopathes whip corals at 3. They are hard to see, aren't they?
Yeah.
Just disappear, even though they're kind of orange. You can zoom in on it if you want. But yeah, we'd like to make our way up the wall when you're ready. Yes, that's a orange whip coral. Stichopathes, it's a type of black coral. Antipatharian.
Mm-hmm.
Kind of a nice indication here of how much morphological diversity you can have at both the colony level and the polyp level within a group. So this is a black coral. We just moved off a black coral that was branched and has those really thin side pinnules and very delicate small polyps where you can see these are much more robust and fleshy polyps.
Yeah.
And the colony grows as just a whip. And this sort of pattern is repeated in the black corals, in the octocorals, in the hydroids. It says something about the flexibility of building a colony, I guess.
There's more stichopathes up here.
Oh, a lot of it, yeah.
You can see the rock wall behind it. It's really flat. And then there's like broken rocks. And they've been here long enough to let stichopathes grow on them. So they look loose, but they're not really moving around much.
This is a nice wall. Be great to stop at that white sponge that's about dead center right now. And coming up, you're a little above it now.
6 o'clock.
Yeah.
It's really tiny.
We've seen a lot of small white sponges. If you see another one,
It's hard to see on this wall.
I know it really is. It's pretty heavily sedimented.
We might have some dead coral rubble on top of that ledge up there. Yeah, a little teeny tiny sponge.
It's great.
You can see his little
Little
oscules.
multiple oscules.
That veining under that, what, subdermal canals is what I think they're called.
Yeah, the canal system. Did you know that sponges can sneeze?
Nuh-uh.
Yeah. So if you, they do. If you take time-lapse videos of these sponges, like over the period of hours, they sort of will inflate and then relatively fast contract and blow stuff out of their...
They like blow their nose.
Yeah, exactly. So it's something that's too slow for us to sort of perceive on the time scales we're used to, but multiple sponges have been documented to do that in the lab.
That's cool. Can you look at the coral rubble at 2 o'clock on top of that ledge? So yeah, I think that's what that is. Just a quick zoom on that on your way up. So that looks like to me is coral rubble. Maybe not. Yeah, it's pretty old. But yeah, it could be like a Lophelia or something similar. Sclartinian. Okay, I'm good, thank you. There's a couple species that look like that. You can see the layering of the rock. It's also really pockmarked, being eroded away. Lots of sediment, silt.
Yeah, we're coming up to about 1,140 meters, so we're up about 20 meters from where we started. And again, this feature estimated to be about 50 meters high. So I'm curious if the lip will have more dense and larger.
There's a nice tunnel there.
Yeah.
Stichopathes is really the only thing. Oh, there's an Antipatharian at 3 o'clock. Brown. Yeah, it's just right of that hole.
Oh, yeah.
It's just a different species Antipatharian.
We'll get a look at that one. It's a third Antipatharian.
Mm-hmm. It's more of a brown branching, sparsely branched.
It's chocolate brown.
It is. Oh, he's got a little crab on him, too. A little squat lobster or something. This looks more like a Tenocidopathes or something. It's really like, they remind me of like pine tree branches.
Mm-hmm.
It's like pine tree branch. So they look like. Oh, that might be an arrow crab. Not sure yet. And then Anthomassus behind it. No, I think he's a squat lobster.
Is that a squat?
Yeah. His one leg looked like a really pointed rostrum, so it's throwing me off.
Nice little Anthomassus-like thing, and then again in this close-up you can see solitary cup coral.
See his little pincher fingers? Good for tweezing.
Mm-hmm.
He's perfect eyebrows.
And really fleshy polyps on this black coral.
Yeah, way different than the other two we were looking at. That's a broken back shrimp.
Tenactopathes.
Also the scale worm on the right.
A little scale worm in the background. Thanks, Roland.
Where is it?
Right here.
Oh,
So,
right,
yeah.
yeah. Nice. We
Yeah,
see that.
Tenocidopathes. I was right. I got it right.
Tenocidopathes.
Yeah.
Very nice.
Yay! I got one.
We see more of the, I think, dead Pharea in there.
Oh, yeah, a little gorgonian under that rock, maybe?
They're under the ledge.
It's tough to see through this. That it's really like marbled, like a brown and a tan.
Mm-hmm.
It's kind of making everything disappear.
See a sponge down in that nook, another little white sponge. Maybe we get a little overhang or another hole, and we can take a zoom and look inside.
Looks like some standing dead coral underneath that ledge too.
Over there, yeah.
There's a different coral here.
There we go.
Or something.
I think that's...
I'm super thrown off by that fairy we saw yesterday that looks so much like a coral from far away, and you get close, and it's totally not what it is. So I'm a little traumatized. What do you think, Alan? Take a guess. Come on, guess. What is it?
I'm thinking this is a really thin...
Quick!
I don't know.
Bamboo coral.
It's a bamboo.
No, it's not. Oh my God.
It's a collateralized. This is a gorgeous...
We are collecting this. Please stop the boat.
Yay.
Oh my God, look how cool.
Oh, Ms. collects a lot.
Look at it.
Oh my gosh.
Wow, it's huge.
Stephanie, I think we collected one of these on the 2017 dive in this area, or the 2018 dive. So I can check. I can try to check the reports, but just so you're aware.
I can't quite...
He said he thinks we collected it 2 years ago.
Oh. We need confirmation, because we should probably collect it if we're not
At least the
absolutely
top.
sure.
At least the top part, right? Where
Yeah.
where it's dead in the
Where
middle.
there's a little gap there.
Yeah. The alien antenna.
Yeah, I'm looking at the dive report from EX 1711, at a depth of 734 meters. We collected this chondrocladia.
Okay, chondrocladia. So we have collected in 2017. Maybe we...
Also, I mean, from the Straits of Florida, it was a little bit further west, and it was at 700 meters.
Yeah, 300 meters
You
shallow.
don't want it. Sure looks like it was this.
Yeah.
So that's pretty good reason to maybe not collect it again then. You want to collect it?
Oh, of course, but you
okay.
know, we don't have to
No.
if we have one already.
Okay, that was really exciting.
Yeah.
I'd like to see, maybe we could scan from top to bottom.
Yeah.
Or bottom to top.
Yeah, pick a direction.
Chris Diaz thinks we should collect it
Okay, done.
anyway.
Done. Yeah, right. Done. We're getting it.
But let's get a good zoom.
At least half of it, the top half. We don't need the whole thing. I don't even know if we can keep the whole thing. It's huge.
Yeah.
Can we get a good zoom on him too before we collect? This is a sponge. This is a
Carnivorous sponge.
carnivorous sponge.
So-
Yeah, we would like to sample it.
But could we image it first from
Yeah.
sort of nice and tight from top to bottom, bottom
yeah,
to top?
super zoom.
So most sponges, they make their living by pumping water through their bodies and capturing small prey items from bacteria or eukaryotes. And this group of sponges has figured out a whole different way to gain their nutrition. They basically capture small crustaceans and then overgrow their bodies. And so that's why they're called
them,
and dissolve them, and that's why they call them carnivorous sponges. And they
Hello. Sorry, Alan, go ahead. It's neat. Look how cool.
It's fantastic. It really just otherworldly kind of organism.
I don't know what these little beady balls are.
Yeah,
Maybe
I was just
reproductive
thinking.
things?
I don't think so.
So do these inflate and deflate?
I don't know if they inflate and deflate. That's a question for a sponge person, Alan.
I do not think so.
I would imagine those little ones have some kind of different function.
Mm-hmm.
But I don't know that either. awesome. So if you pan down a little bit, right in the middle, there's like a dead spot. That's like a perfect place, I think, to give it a good snip.
So, they do separate gametes, one from the other, and what Chris Diaz is saying that she thinks the large ones are what contain the sperm, and then what we see in the smaller balls are where the eggs have been sequestered, and so presumably sperm would be released from the large balls, and then other.
Look at that one was merged. See here. Yeah, I would think here, but it depends on how much space you guys have, and I'm not really good at judging that, so you can get less if you need to.
Yeah, and these are really. I was reading the other day about these guys, at least the species that have been studied. The sperm don't have little tails on them, so they actually make little sperm bundles. And when those are caught by another sponge, they come apart, and then the sperm burrow through the sponge tissue to fertilize the eggs.
Yeah, and then lasers on it before we snip it too would be great. Awesome. This one's probably been here for a really long time.
Yeah.
And then just take a little piece.
Yeah, George Matsumoto is chiming in as well that the upright branches produce packets of sperm, and those are released into the currents and are captured in the branches of nearby sponges. And then, as I was saying, the sperm works its way from the packets into the host sponge.
You got some worms in there.
Nice. Is that above or below where we're collecting?
That's below.
Oh. Okay.
Yeah, it's pretty close to the bottom.
Yeah. Now we'll just take it where we're planning on taking. I believe that some of the. And then once the eggs are fertilized and mature, that's when they swell up, forming the bulges partway up the host.
Is that too big? Do you need to go just like higher than that? You're probably going to need your clippers. That center stem should be pretty stiff. Good. This is sample number 4. Correct. Yeah, you're going to need to snap it with your, yeah, try not to pull the whole thing up. But you're going to have to cut it with your clippers. It looks like you missed it. I think can you open the jaw back up and put it deeper into the snipper part? Oh, was it stuck? Okay. Okay. Ooh. There you go. Perfect. There you go. Good job. Yay!
Oh,
Secondary filaments. So she's talking about the secondary filaments are swollen along the length and not terminated in place like that.
ah,
So this one is actually
we are just completing our pilot transfer. Okay, I think we're back and ready to proceed. See some more of that Stichopathes off in the distance to the left a little bit. I'm sort of curious what that white, is that a white sponge in front of us? Yeah. Yeah. Oh, maybe just exposed rock. It's a nothing. But boy, when you zoom in, you can see, look at all those different demo sponges. It's just the wall is covered with them. We got some good close-ups of those before. Mm-hmm. And we're about 20 meters from the top of this or so. I think actually it would probably be a good time to when we get up closer to the wall to take a next DNA the water sample, because we're about halfway up, I would say, in the ballpark of. I think we can do one in the middle of the wall, so we'll also still have two more for the top. Great. See a little. It'd be great to get comfortably near. Yeah, avoid stirring up the dust, thanks. Although there's DNA in that as well, but just to keep it consistent. Yeah. Ready.
See it on stream 3.
Great. Thank you. We could take a rock sample. What do we have though at about 8 o'clock now? Is there yellow in there? Yeah. hmm So we have that yellow farreid again with it looks like a piece of Lophelia or other scleractinian rubble. Maybe we'll find them live at the top. And a nice little squat lobster on the sand there just to the left. Light white pink. And you can see in and amongst the rubble lots of more of these demo sponges that are encrusting on pretty much all the hard surfaces that we've been able to take a close look at. You can see where some of these pieces have tumbled down and are just perched. That looks pretty precarious there on the top left. I think that the white, less eroded stuff might be most helpful, but yeah, that looks great. It is kind of eroded out pretty bad. Nice little ophiuroid there. Let's see if we can find a more smaller piece. You'd like more dense piece. Yeah. I don't suppose it's absolutely necessary to do it here anyway if we don't see something, but we look under the undersides of some of these ledges to see if we see anything small that's sort of growing down from them. Yeah. 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 That's interesting. That looks like more coral. Oh, is that? No, I don't think it's. Can't tell if it's alive or not. You see the single solitary cup coral, but then this colonial form too. Thanks. Mm-hmm. What's moving? Oh, there's, yes, that may be the mama octopod. No, I think it is moving in there, right? It's hard to get in there. Huh. And it's just an illusion. Looks like we can see some more of the blue sponge, which I think we sampled the other day. It might be different or it could be the same. It wouldn't be bad to have a snap zoom on one of these blue guys to see how it compares to what we collected the other day. The sample of the blue sponge that we did collect, one of the interesting things happened when we fixed it. All the blue pigment was soluble with ethanol, and it went into the ethanol, and we've been seeing how stable it is. And 2 days later, the ethanol is still very blue. Oh, no, I think that is not the same sponge. It's certainly not as strikingly blue as the one we had sampled before. Although it has, I guess it's possibly the same. Yeah, I think when we get up this close, it does look very much like what we collected before. Thanks for that. You know,
Sorry, Alan, go ahead.
No, we're just curious about getting a rock sample at some point for sure. But I was going to say that, because we were able to collect that blue sponge, we'll be able to identify it, and then it's something that will be, coming back through here, we'll be able to really pick out how common it is and put a name to it.
Yeah, and just skim past them.
Mm-hmm. Yeah.
Then they become part of the background. Beautiful blue color, though. There's a rocky place.
And we saw an interesting coral while you were under a ledge called Cladicora, maybe.
Yeah,
John Reed thought.
Mm-hmm.
Yeah.
Look at these big broken ones. They look like that typical, old reef.
Yeah.
I wonder if this was old barrier reef or something when the sea level rise was lower.
Yes, when the sea level was lower and probably this bottom was higher.
Mm-hmm. I see a lot of that dead coral rubble.
Yeah, I'm wondering if we're going to see live coral at the top of this.
There's something interesting, maybe man-made. It's just coming into center now from the right. If you could just kind of zoom in a bit. This thing, yeah. What is that? That's weird.
Carbonate.
Think so? It's very consistent. All right. No idea. If it's human-made or looks like a tube or something. A dead coral, solitary cup coral on the ground.
Mm-hmm.
That's different.
Oh, maybe that's a fossil.
Looks like the upside down agaricid almost, or
If it's a table coral tips
Yeah.
upside down.
need to flip it over.
Looks like a very large foothold.
Yeah.
Mm-hmm.
Something.
So the shallow water corals will grow like that, real robust. What am I thinking? A Carapra palmata, maybe, or a real big agaricid.
Yeah. Just diving on a fossil reef here.
Mm-hmm. Could be, yeah. That's what it looks like.
It's always the best stuff lives under those. So I always want to, when I'm snorkeling,
Go peek under.
peek under these large flat pieces of rubble. Yeah, let's check out that anemone. I'm all for it. It's nice next to a Stickopathes.
Yep, and flytrap anemone. There's another Faria. Looks like standing dead coral, too. Cladocoras, maybe.
I think that's quite beautiful, striking.
Oh, I can't. Flytrap, thank you. My brain wasn't working there.
So that's a flytrap anemone?
I think
as well?
so, yeah. I bet if we look underneath the one edge, it would be really bumpy.
Mm.
And then we got
Lots of Faria. Oh, look at that. Nice. Really great view of this rock. Yep. That looks like a bivalve. And that white sponge to the right, I love it.
A couple of them below it. Alan, can you spell Faria for me?
Farrea. Faria. What do we have in the water column there? Yeah, let's take a look.
What is that?
I think it's a siphonophore, right? Yeah.
It's gotta be.
Oh, it's a benthic siphonophore. Nice.
This one's super calm and chill. Remember the other one who was like losing it.
Yep, but this. Let's get it. He is floating up. He's released from the bottom. Is he still connected? When you can see his tentacles coming out with its side branches, the tentilla. Those are the parts that are specialized to catch its food. And it looks like it's also losing some just in this. You can see how delicate they are, and you imagine if we collected it, it'd just come apart in hundreds of pieces.
Yeah, and sting everybody.
And sting everybody.
Looks like a little balloon.
Yeah. And again, this is the animal that holds on to the bottom and then deploys its tentacles and catches organisms floating by close to the bottom.
10-centimeter bivalve shell. Oh, we got lots of Faria here with lots of zoanthids on
Lots of zoanthids on this
Barely
one.
infested.
And those are different zoanthids, aren't they?
Not sure. Maybe.
I think they don't look the same.
This one looks. These look yellow.
Yeah, these
I don't
look.
think the other one looked yellow, did they?
We could see their central sort of gut region in this. We'll take a. It'd be great to take a look at this.
Maybe some cladorhizids on that rock. I see the little
drooping down.
Yeah, the toothy things.
We'll have to take a close look at that as well. I'm loving this. Even has the great.
It's interesting.
Yeah.
Lots of zoanthids.
It's just.
Look, you can see in that one. You see into the hole in that one?
Yeah.
This one looks pink.
Yeah. One of the zoanthids?
Yeah. Do you see the pink?
Mm-hmm. Mm-hmm. All right.
Cool.
It's so cool. You can see how delicate this sponge is.
If you think it's a different zoanthid, is it worth getting?
I think it's. I'm sure it's a different zoanthid. Well, as sure as.
Looks like
it really looks different to me. It does not have the same gestalt at all.
And then I'm seeing. I think these are cladorhizid sponges.
Yeah.
You also have a gastropod.
Gastropod, a snail there. Mm-hmm. To the right. Yeah. Do you think we could have time to grab another Pharea plus zoanthid?
It's up to the pilots, right?
That's great. Let's do it again. I know somebody who's going to be excited about. Tim Swain is the person who mentioned that there's a lot of undescribed zoanthid.
We should grab a cladorhizid too,
yeah?
Okay, while we're here.
Yes, please. Those spiky balls also would appear to be in the cladorhizidae.
Oh, dive 10 or dive 11? Or dive 11. And the last one I have is sample 4. Is this going to be 5, or did you get?
No,
something?
we got the water.
Okay.
We got the mid wall water.
So 6.
Yep.
Yes.
Yes. And then we're also interested in the pom-pom things.
Yeah.
Yeah.
You could probably suck this one up too, if that's what you want to do. It's going to be really, really fine.
I think it's a little bigger though, right? I don't want to get it stuck.
Yeah.
So those cladorhizids have never been collected by Okeanos Explorer, so we got some very enthusiastic chat participants saying, yes, please. Maybe we can get more than one of those guys.
Yeah, a couple of them.
Yeah.
Poop balls.
We'll worry about the yellow collection first.
Did you see if the Faria tissue was yellow? The other one was, right?
The other one was, yeah.
I think the tissue on this one's white.
Mm-hmm. But the zoanthids are yellow.
Mm-hmm. Yeah, it's gonna be a really crisp and fragile sponge. So if you do use your suction hose, you should just need to scrape it a little bit at the base, and it should just pop right off and float right in.
Yeah.
If you were to grab it with your claw, it would probably just crush.
It would be great to get the squat lobster at the same time if it comes in. Yeah, because of the association, and people have requested them, but they're often difficult to get.
Yeah. My concern is that you'd have to turn the suction on too high and destroy the little faria. Please. Yeah.
Great. Sounds good. Yeah.
And I think the small spiky balls will be good on the suction sampler too, so maybe we can knock out 3 samples right here.
Yeah. Looks like a great spot. Mm-hmm. Sounds great. Like these associations because it's, more
More data?
more data.
Oh, John was saying this is the Mitchell escarpment. Did we get that out?
No, I didn't mention it yet. I did see that, but Mitchell escarpment.
Mm-hmm.
pom-poms don't have a name yet. Like, they're definitely undescribed.
Yeah, if you just scrape the base, it'll pop off. Oh. This is stuck.
I wonder if there's like a...
It's snagged on something. Yeah, tap it on something.
I bet it's got like a little lip.
Mm-hmm.
It's great to watch. I never ceases to amaze me.
That's being stubborn, huh?
Well, it's just got a little, just a little over the edge.
Mm-hmm. Oh, yeah.
Oh, yeah, definitely.
There you go.
There you go. Nice.
So number
Well
2.
done.
2.
That was 6B.
Mm-hmm.
Mm-hmm. That'd be great. And this is going to be an undescribed species of Abyssocladia.
Okay. So this is going to be sample B.
Mm-hmm.
There's some on top up there? Or no? Okay. Sure.
Yeah.
Okay.
Mm-hmm.
Great.
That's great. Thank you, pilot.
These should be fairly similar, like crispy. Yeah, should pop right off. That's fine. We'll deal with it.
That's right. Unintentional bycatch. Wow. Come on, guy, pop off.
There goes one.
You got one?
Nice. Ooh.
It's almost off. Sort of deflated a little bit, but still, make a great DNA
I was
sample.
going to say it's going in the DNA jar.
There we go.
Thank you. If you don't want to keep going, we're fine with two. You don't have to. If it's a battle, it's a battle. Just poked his arms out.
Yeah. Pom
I mean,
pom.
you don't have to beat it up too much. It's fine if you only get two. Oh, we got this primnoid too coming in. Switch buckets.
Well, we don't want to. Well, it can go into another one, but or it can just go loose.
Yeah, let it go. Maybe drop the suction.
Yeah, if we get the coral, that would be great. We can add that to our collections for sure. That's all right.
happy with he's way under there. It to us, yeah, if you can get it in a different bucket. Okay. I don't foresee him getting caught, but...
Oh.
What?
I just got in the text. They're quite interested in the coral that went away. It might be something that Steve Cairns just described. Okay. Sorry, I was a little slow to get to the chat. Squat lobster.
Like a lobster.
I guess they're hard to get out. Run into, yeah. No problem. But if that coral is possible to pick up, we do have a request for it. Thanks, Pilot. Mm-hmm.
What was in 7? Oh, 6 was the sponge, and then 7 was the little poof balls, and we didn't get anything else, right? Yes. Yes. Oh, what went in 8 that I missed? What went in 3?
Oh no, they'll just be 7.
7. Sorry. Yeah. No, they're going to be one sample.
Always best.
Yeah. 2 individuals.
Now, do you see the coral?
I don't see the coral anymore. It fell down the wall.
No, I think these guys, the pilots and their sharp eyes. Yep, that's it. You can see the little extra puffball stuck to it. No coral left behind.
Primnoid? Is that what they thought this was?
That says a planar chrysogorgid.
A chrysogorgid?
Might be the new taxon Steve Cairns published on this year. So this would be 8B, new or unusual morphotype, I think.
Uh-huh. Mm-hmm. Nice.
Mm-hmm. That's great. Yes. You might as well collect the rest of this wall.
Sit here for 10 more minutes. So this is going to be 8.
Yeah, 8B. Starboard inner is the probable destination. I think we're going to make it to the top of this wall.
I hope so. What time is it? 1 o'clock? What's our off-bottom time? 3:30?
Yeah.
I don't think we have far to go, do we? 10 more meters?
Yeah, 10 or so, and then we can traverse it.
Oh, yeah, we'll be fine. Good job. Thank you.
Great.
All right, that was sample 8B. Chrysogorgia.
Mm-hmm. Well, Chrysogorgid.
Yeah, Chrysogorgidae.
Big thumbs up from everyone. The benthic biological world says thank you.
Ooh, look at swimming in the
Oh,
water
was that
walker.
another ice pod?
Mm-hmm.
I'll make note of that. Little munopsid. It's always so funny when we're looking at this view. It almost seems like there's nothing, right?
Yeah, and then
But
you get close.
every time we start, say, "Oh, let's look
Yeah.
at this," and then
It's like 3,000 things pop out.
yeah, it just keeps amazing me over and over.
It's nice scintillation holes in front of us.
Mm-hmm.
Scintillation holes.
We were looking inside one of those holes.
Did they have specify the rock type they were looking at?
No,
or?
I got nothing from the chat on geological samples when I
Okay.
was trying to ask what would potentially be useful.
In the sinkhole, they said the less eroded, the better.
Mm-hmm. But all these
The
seem
lighter,
pretty.
the better. Yeah, these are big honking rocks.
What's that? Red shrimp?
This brown thing, I don't know what that is. There's a neat anemone or coral or something. Yeah, it looks like old reef or consolidated sandy. Yeah, it doesn't look like that clay that I was expecting. It's the same color.
Right, but it's
But
definitely,
it's definitely
more consolidated,
yeah.
more.
More of a rock and less of a, less of a dissolvable.
Mm-hmm. There's that yellow farreid, urated, same glass sponge.
It's neat, like steps up here. There's a coral.
Yeah.
Yellow stylaster. It looks like what he's growing on was coral at one point.
No.
No?
Is that stylaster? Does it look like...
Madrepora?
Yeah, I think that's more of a scleractinian type.
Yeah, that looks like Anelopsamia, a scleractinian,
Anelopsamia.
which I will write down in the chat.
It's very fine.
It is. Well, that's neat. Nice.
I probably would have gone more with Matrices or Madrepora. It's hard to tell. All right.
So, yeah, don't trust my genus name on that. I was just referring to a genus that has a planar growth form that's a scleractinian. But Steve Oskevich still thinks it's a stylaster. I don't
Really?
know.
Okay.
But I thought I could see large polyps on it.
Yeah, they had the really fine. They're not
Oh.
hydroids. They're not little hydroid hairs.
Steve says they're definitely stylasterid. Huh. I thought I saw...
So it looks like a solitary cup coral. It's dead.
Mm-hmm.
Yeah, and I didn't get a close enough look when we were zooming in.
Well, I trust Steve, but we're going to have to look at it as well just to verify.
A pretty old dead coral polyp there.
I did want to note it in general, as you're passing along the wall here, and we're really not seeing much on this wall, but we are seeing a bunch of rubble, an indication that it's pretty friable. This old reef is breaking up, which is why we don't see any large colonies growing on it. The substrate isn't stable enough here to grow on, so instead it looks like they're growing on the rubble down below.
It's pretty rugose. Lots of hidey holes.
In the canyons that we dove on up in the northeast, these hidey holes would very commonly have octopus in them,
Oh, nice.
rooting their eggs.
Sweet.
It wasn't quite, it wasn't full reefal material like this, but it was carbonates, I believe.
That looks like a dead Cladicora. At 6 o'clock. Yep, crinoids at noon. Ten-armed. Really long, the end of each arm has this really long pinnule. So, Anelcedometra, maybe? There's a big burrow there.
Wow, look at that.
There's another one of those sponges.
Yeah, it's really growing large. We have the lasers to...
This one, coated in zoanthids, right?
Yeah, it's sporting a lot of zoanthids. It's...
Like almost can't even see the sponge anymore.
Right? Yeah.
Yeah, I was going to more zoanthid than sponge.
This one looks like both the zoanthid and the tissue of the sponge are yellow.
Well, one wonders if the sponge is still alive.
Right.
Really nice.
So that's probably the same species we just picked up.
Mm-hmm. This looks very similar.
Now, I'm looking forward to what Tim Swain says about the last one, but, you know, when I look at this, there is such a thing as a colonial anemone. I can't remember the name right now, but just there's something about the oral disc and the tissue here. I just wonder.
Okay.
Isn't that what a zoanthid is?
Well, it's he means an actiniaria.
Oh, okay.
As
Oh, like a
opposed-
colonial actinaria. Okay,
Yeah.
got it.
Right, yeah, exactly.
Okay.
There aren't very many, but there are some. Sinanthus, I think that's the name of the genus, Sinanthus. I wonder, Alan, do you know, do the double layers of tentacles there tell us anything about whether it's zoanthinarian or...
I think that's consistent with it being the zoantharian.
Okay, great. Thanks.
See, with the...
Yeah. Some point up, some point down.
Yep. Every other one goes up and down.
All right, we're good whenever you guys are.
There's another large colony to the left, something different on this big rock.
I think it's right. There it is.
Oh, it's the same. I just didn't.
So this
I
one
didn't
doesn't-
realize it farther. Yeah.
It's a fairy with no anemones? You see
Yeah.
anemones on it?
But, snap zoom might be nice for comparison.
And then no anemones.
Nice little hermit crab on top of that guy. Oh no, there's a zoanthids
Oh, there's
in
a
there.
couple, yeah. 3 or 4.
But
But
much
definitely.
more, and we can see a much better, clear view of what the growth form of this
Yeah.
sponge is. So this is going to be really nice.
It looks almost mechanical,
Yeah,
like a machine.
1, 2,
Like
3,
consistent.
like it's twisting.
And the top is white, the mid part is yellow.
Yeah, we've seen that consistently too, where the yellow pigment is lower down in the sponge, and then presumably the growth edge, fresher, younger sponge is.
It could. Some kind of chemical deterrent or
Or it could just
You
pick
picking 'em
things
off,
off. Yeah.
keeping it from settling out.
There we go.
Yeah, there we go.
Ooh, that's nice. Good spot.
Little gray guys.
Boy, that is a very young coral. Or else, maybe I suppose it's the tip of a coral that's hidden behind the rock.
Yeah.
Could be.
I'll take a look at the top of this.
A lot of the times when you're in the Keys, they look like they've used like old reef as building material. Like it looks like pieces of cut coral. And I would imagine that they're getting it out of some place like this. Not sure where that building material came from. It looks a lot like this. Oh, I think we have another cladorizid. Yeah. Can we zoom in on that stick in front of us?
Yeah.
Yeah.
Yes.
Pretty cool. This
Chondrocladia
one's
again.
a lot shorter.
Curious what's going on the bottom of it too.
Another squid. What's that green stuff? Probably human debris, maybe? Some rope? Yeah, 7. Yeah, it's like old rope.
Mm-hmm.
And the standing dead coral.
More squid ink.
Squid ink. We've probably another 5 meters to go to get to the top. Ooh, is that a
Just on the ledge there in front of you?
chitin, maybe? They have deep water chitins?
They do, but can we look center?
Just a rock or something like that.
Not a rockfish,
Oh,
just
no,
a
that's
rock. Yeah. Ooh, nice. Guess that's
Hylanema?
yeah.
Something similar?
Hyalonema. Curious what will be living on its stalk.
Looks like it could have zoanthids around it.
No.
Not yet.
Actually, this looks quite a bit different morphology than the other recent hyalonemas that we did see. This has a much more open top to it. Like a goblet.
Mm-hmm. Like a wine, like a nice wine glass. Mermaid's wine
As
glass.
usual.
What, Scott?
I said zoanthids on the stock as usual.
Mm-hmm.
Yep.
Look at this little hermit.
Great hermit.
Teeny tiny. He's itty bitty.
And we'll get a close-up look at those zoanthids as well, but I think they're going to be different. They look quite a bit different textured than the ones. I don't think so. Yeah, this is great. That's an interesting looking squat lobster. Yeah, it really blends in great. This is Chris Diaz reports this is Hyalinema CF Conqueror, described from Brazilian deep waters. So I wonder, could we make a collection of this with the zoanthids on the bottom? This would be another, and maybe we get lucky and get that crab. It's turning into the sponge zoantharian dive. I don't believe we have. It'd be great to get.
Your squat lobster looks like another species of Munidopsis, but which one, I don't know. You can tell because it's blind.
Thank you, Mary. Munidopsis. Yes, please. So that fish that came by was a halosaur. Thank you, George Matsumoto. Thought it looked. Was it a juvenile? I'm curious. If you're listening. I wonder if that is Nematocarcinus again. The red shrimp, very long legs. Yes, we have confirmation that that was Nematocarcinus. Yeah, a little rhodalid on the sand. That's dandelion siphonophore.
I don't know if you intended to try to catch that squat lobster, but I should warn you that they escape by swimming backward like a crawdad.
Yeah. I think it would be no, it would be nice to concentrate on the squat lobster. The yeah. Yes. And then maybe we're going to reassess the sponge. Nice. Siphonophore dropping by. Yeah, same one. There's been a benthic siphonophore floating by. We're actually going to try to collect a squat lobster off the stock here, and we were considering collecting this sponge with the zoanthids, also for the Tim Swain research. Wow.
Cool.
Yeah.
Now, is it the same species we saw earlier? It's just that it was really active?
Port inner accidental? Oh. Gotcha. So I...
He sampled himself and then took off.
He went in and out?
Yeah, the siphonophore went into the sample bucket and then left.
All right.
So we're getting the Hylanema?
No, I think we've decided in the chat after some discussion to not go for the sponge and zoanthid because Scott has shared that already. That exact same. Yes, please. I'm going to give it a go. Thank you. Right. And this is Munidopsis.
And it would be 9 if we catch it?
B9, yep. And I think it would be going for Canister 3 if it collected.
Jar 4.
Oh, thank you.
Mm-hmm.
Well, that isn't really what they intended to do.
Well, that's great.
Yep. No, this is Okeanos Explorer.
Mary, can you mute your phone, please?
NOAA, they don't get any money from National Geographic.
No, I don't think so.
Sorry, go ahead. What were you saying? Yes. Okay. Yeah, you can if you need to.
Yeah.
I mean, we can lose a bucket if we have it. Oh. Yeah, if you can. If you're worried it's not going to
Go in.
come in otherwise,
Yep.
you just give it, turn the suction on bucket 5 and see.
That's great. Thanks a lot for that. Oh.
Yeah, it's fine. Whatever comes out, comes out.
Absolutely. And
We'll process whatever's there.
yeah, I may sequence the genome of that sponge.
Yeah, do it. Do it.
Do it. I think Hyalonema conqueror deserves a genome.
Mm-hmm. If you want to look at them, we're fine.
Yeah.
But he looks pretty mad.
Oh.
He's threatening the right?
Angry.
It's getting pretty dark.
Maybe.
Awesome.
Yeah, that sounds great. Follow it northeast. We want really at the top.
Wherever the most biota is, in my opinion. But,
Yeah, which is probably going to be the edge of the ridge, right?
Mm-hmm. We have until 3 or 3:30. Is that right? Thank you. There's not much up here. Looks pretty smooth. Yeah, probably. Are you? Okay. Yeah. Okay. Mm-hmm. Yeah, that sounds good to me. Just try and keep the biota heavy wherever we can. Yeah, it looks like... Mm-hmm. Yeah. That looks good. That looks like there's some tall stuff poking up on the top of this, too. Get a little closer. A little squid. So it's like a almost 45 or 55 degree slope in parts. Here, the bottom rock is really smooth, and then it seems to break off into these jagged edges. And then there's these larger boulder-like size, really jagged black rocks that kind of almost look like they're sitting on top. Oh yeah, we can we do a Niskin up here? Well, here's fine. Yeah, this is fine. That's going to be 10. Thank you. That's fine. Yeah, the tall guy, probably. These rocks are really weird. It's almost like they've been, like, dropped here. I don't know where they would have come from, though. There's a standing coral there at 3. I'm not even sure if it's dead. It looks like it might be. There's a big crazy fan, yeah. So purple on the top. Oh, I did that already.
Oh.
This looks like our first purple clavulariid, which is kind of like a mat-growing or ribbon-growing octocoral that overgrows other corals and skeleton rock.
What's the purple?
Clavularia.
Clavularia.
This, I think, is the species, but I like to call it the royal purple octocoral.
Thank you.
That's really beautiful.
Mm-hmm. There's also a hard coral underneath it.
Hydroids to the left.
Tons of stuff on this guy. Hermit crabs. So yeah, this is maybe Inelopsamia or something. That looks like the same coral we saw earlier.
Mm-hmm.
It's Lophelia-esque, but I don't think it's Lophelia. It's got a yellow tinge to it.
You can tell it's a mysid because it doesn't have any obvious abdominal appendages. The true shrimp do. You can tell it's a mysid because
Cool.
it doesn't have any obvious abdominal appendages.
It almost has like feather-like swimmerettes. What I'm thinking I'm seeing here.
That big fan that you're looking at, that's behind the mysid, the opossum shrimp. Candidella imbricata, and again, you can see several places where there's thickened white parts in the branch are where polychaetes are, the scale worms, are living in their little tunnels.
Nice.
Wow, this is a great spot. So when you collected that last water sample, were you near?
We were at the top here, so
Mm-hmm.
we were trying to get on top. Looks like whatever this is growing on was pretty hefty at one point. Giant fan or something. How about this yellow one at 6 o'clock? That's different. Yeah, at the bottom.
That looks like an acanthogorgia.
Mm-hmm.
Acanthogorgia.
Mm-hmm. Yeah, and it looks, I'm pretty sure this is an Elopsamia, but above it, the hard coral above it.
Got those
Couple
barnacles, yeah.
Little shrimps in there. And then Cametulid crinoids.
Well, that's a nice, on the back side. Can we zoom in on that little colony?
Where are you looking?
We're just behind the stalk.
Oh.
Sort of white and
Yeah,
fine.
what is that? That looks like chrysogorgid over there.
Yeah, behind that.
That might be our
But
last sample.
This little guy.
Hey, Mary, I know you're on the phone. Hope you're still there. Can you get a peek at this chrysogorgid with the squat lobsters and tell me if those are what you're interested in? That we need data on?
It sure is. That's the genus Eurotychus.
Okay. Hey, pilots, can we collect this gorgonian with the crabs on it? Yeah.
These fellows tend to be rather slow-moving, so you have a better chance of getting them.
Yeah, good.
And
So that's going to be...
This is one I would
Go ahead, Scott.
definitely clip Chrysogorgia colony to
Yeah.
get that squat lobster.
Yeah, we're going to need to clip...
As opposed
Use your claw to clip that. And put it in a bio box, please.
And I think one of the things Mary can speak to here is my understanding is a lot of these chirostylids have a host-specific relationship, so it's really good to know what is the coral that it was collected on.
Mm-hmm. Yeah, we're gonna grab the coral and the
squat lobster.
squat lobsters. Just get them all in a chunk.
And is it chirostelid? What's the name of the squat lobster?
Chirostelidae? I don't know if Mary has a better name than that. But
I believe is the family. Or
Well-
not sure if it's the family or higher.
Would you say Unimopsis or something? Mary?
The family is
Go,
So-
Mary.
The family is Chirostylidae, but the genus is Eurotychus. Uroptychus.
Thank you.
And most of the hosts have never been reported, except maybe the family. So it's quite possible it is host-specific, but we don't know what the host is.
Awesome. So this is a good way to expand our knowledge. U-R-O-P-T.
And this, we can get more great look at this clavularia. I just think that color is really stunning.
Oh no, purple.
Mm-hmm.
Super purple. You can see, look, the one it looks like it's got its polyps in.
Mm-hmm. So it's just encrusting on the old stock, right?
I don't know. It looks like it.
Yeah, I think that's correct, Alan. It may be able to form a form of skeleton of its own, but it's typically stoloniferous or mat-growing type cruster.
Yeah, it's neat to see it with the polyps in and the polyps out. Get that comparison. You can see how it's just like a sleeve down this old dead arm. And I hope we've mentioned the nice white hexactinellid in the middle there.
We have.
There's so much to see on this.
Oh no, we haven't talked about the hexactinellid.
Well,
We have now.
It's a hexactinellid.
Yeah. It's just his own little community here. There he is, a worm.
Ooh, he's cool.
He's shiny.
Did you ask if you can grab that? Darn.
He looks like he's got rainbow legs.
But you knew I wanted to.
So if you lost place of the chrysogorgid, it's actually on the lower right behind that. Yeah.
This whole, I don't know.
Dive 11, sample 11B. I think we have one more box left. Oh. Yeah. Yeah.
Nice. A pair. Go ahead. Or did someone hang up?
Hey guys, this is John
Hey,
Reed.
John.
Hey, that hard coral that you were looking at a few minutes ago.
Yeah.
I mean, I couldn't really get a good look, but possibly Salinas milia,
Oh, Salinas.
if not in Alabama. They both occur out here in deeper water, deeper than Lophelia usually occurs.
Yeah, I didn't think it was
Salinas
Lophelia. It was definitely like,
No,
yeah, definitely differently shaped.
no, it's one of those. It's either in Alapsami or Salinas milia. Salinas milia is bifurcated at the tip. I couldn't see if it was, but you got it. If you get a piece of it, might be good too.
I don't know if we have any buckets left. Oh, maybe we can do port outer. We can try. We'll see. I can definitely get a better look at it.
Yeah, that's
So if
a
you
good
just hang
idea.
out and watch, we can go and get a good shot of it.
How do you spell Salinas milia?
Oh, yeah, I don't. Working on that.
I'll look it up. I've
That's
got
what
it.
I was doing.
Yes.
Yeah. But they're not going to come off that chrysogorgid, so your best bet is to snip the chrysogorgid and put the whole thing in a bucket. Okay. I think it's Selienious. Oh. Oh, okay. I got it.
Soleno smilia.
collecting fools today. No, I know, right?
It's an exciting day.
Yeah, it's lots of stuff here. Lots of new things. I was expecting a
suppose
sand dive, too.
Advantageous, I know, that we can sleep in the morning.
Yes. Yay!
Colors.
Ooh. There you go, you got it.
Nice, nice, nice.
Even got a little piece of coral rubble underneath. We lose one of them? All right. Sorry. Is he in the bucket? No!
No!
He disappeared. Do you want to keep the coral?
Yeah, we might as well.
Yeah,
we
just put it in the
plucked
bucket.
So, yes. That's all right.
Mary, we tried.
I heard her in the background.
I
Did
think
you
it
hear
was
her?
me.
Oh, was that you?
Yeah.
Oh. What's the echo? Okay, we got the Chrysogorgia.
No, it's fine.
No.
We'll try again. Think they'll find each other again? Find a new Chrysogorgia to hang out on?
Yes, absolutely.
After seeing all those squids, I'm not very optimistic.
Oh. If we didn't get them, the cephalopods will.
Yep.
Well,
Yeah,
Sounds
keep going.
good. Oh, did you want to look at the scleractinian?
Oh, yes, please. Can you swing back to that large coral thing we were looking at?
Sorry, there's...
And just take a peek at one of the corals in the middle.
Check the ID again.
It's the yellow hard coral right in the center right now that everything's kind of growing off of. There was just a question about what species it was. John wanted a sample, but I feel like if we get it off this, we're going to have to process a billion associates. Yeah, so that's all overgrown. What moved?
Yeah. Something like closed a trapdoor.
Yeah. Weird. There's a good pink color right here. That looks like it's
alive.
Yeah,
and
alive.
Can see the skeleton a little bit better.
So that's at 3 o'clock. Yeah. Yeah, kind of in the background a little bit. There you go. Let's get a good shot of that, and then we can get it ID'd maybe. All right. All right, John, if you're watching. Oh, that look Clarisid's on there.
Yeah.
So it's this pink one that's right off screen at 3. This is the one we're after. Yeah. It's really rigid. Okay.
Thank you, pilot.
Thanks, guys. Yeah. There's the crab. Yeah. We stole his home. Sorry, buddy. Oops. Okay, we're good whenever you guys are. I'm not sure what that is.
Is that the planar chrysogorgia?
Yeah, are you referring to the thing that's on the lower left?
Yeah.
Yes.
Yeah, I think that's what we collected earlier. I mean, the one that was suctioned.
Mm-hmm. Yep, sea star. Oh, he's upside down.
Yeah, you don't get that view of a sea star off too often. A little ctenophore just went by. It's like a trapeze artist.
Mm-hmm, right.
Who's in trouble?
Yeah. And eating while performing.
Is that same planar chrysogorgid?
Yeah, it looks similar.
Did you not hear me say that that's the one that we collected earlier?
Oh, I missed that. Sorry, Scott.
Yeah, then this was a different one we were looking at. But yeah, it looks the same as that.
Oh, I'm sorry.
That's okay.
My screen is frozen, so I'm not looking at what you are.
Okay.
Oh, we're just looking at a star that appears to be climbed out on top of it. Presumably
Yeah,
feeding.
I'm talking about something completely different. I apologize.
It's all right.
Christopher Moss says he's going to bet that it's a Circeaster.
But now I see what you're looking at. There sure aren't many polyps left on this thing.
No, just some out to the left on this colony.
Yeah, I see a few there. And that's consistent with the Pleurogorgia type of Chrysogorgid. So that's consistent with what I thought earlier, that it was kind of a special planar Chrysogorgid.
Actually, Chris Ma has updated his identification to a name I hadn't heard, Floriaster Maya. See, I guess the zoom really helped.
Mm-hmm.
You know, what's interesting here, I'm not sure what Chris is saying, but this skeleton looks like it's been denuded for a very long period of time. Longer, I would think, than this sea star was here, but who knows. So I wonder if the sea stars may be feeding on other overgrowth on the coral.
He also doesn't look like he has his mouth out. It's like he's more like stuck to the coral with just his two legs. Or arms.
The stomach is not extended.
Mm-hmm.
Could he be climbing back down after eating?
Yeah, he could be going back somewhere else.
I mean, it's possible. What I'm looking at is the rest of the skeleton where there's no tissue. Looks like it's been exposed for a very long period of time.
Yeah.
There's all kinds of stuff that's stuck to it. But, you know, maybe in this environment with a lot of marine snow and a lot of this stuff drifting off of the shelf, maybe that's what it looks like, you know, just in a few months or a year.
Yeah.
That was great footage.
Sticopathes.
Elix going by.
This definitely looks like a old reef. Fossilized reef or something.
Oh yeah, good eyes.
Another Hylanema too. Right at the top.
Mm-hmm. Hylanema conqueror, or CF conqueror.
Or that purple Gorgonian, purple encrusting Gorgonian maybe? Really bright.
The royal purple?
Yeah. Can you look at the coral that is at 9 o'clock? There's a couple of them. There's a yellow and there's a white. But it's just 9 o'clock of where your laser should be. Right. There's a white one right there, center screen. Yeah, that's what I'm looking for. Yeah.
Pom-poms?
So, yeah, more cladorhizids. So here's that same scleractinian we saw earlier. And a lot of the dead coral looks similar to this. So John was thinking Solanus milia, possibly. Do we have one? We have one bucket left. You think you guys can grab a piece of that? That coral, yeah, right there. The hard coral.
There's the
another
Chrysogorgid with the... you again.
We'll definitely see Chrysogorgid again. I would go for this hard coral. No. Yeah, if you get it, great. That's fine.
Yeah, if you get it, great. That's fine.
So this collection is to confirm the identity?
Mm-hmm. It's also, there's dead coral from it everywhere, so at some point it was probably pretty abundant here.
This is going to be 12B, right?
Mm-hmm.
There may be a Nephtheid just to the left of this scleractinian. We're just having a quick pilot change before we finish this up collection and the rest of this transect along. Oh,
Maybe another half hour left or so?
yeah. I'm trying to what was the name of this region? This scarp?
Yeah.
John
I know.
Reed
I
told
know.
us.
He texted me. Hold on. It's in my text messages. Hang on. He said Mitchell's Escarpment. Mitchell Escarpment.
Mitchell Escarp. Gonna put that in my notes. We were scrambling to get this site.
Is it okay to use the suction hose since we had that thing kind of maybe covering? We worried about the other sample being stuck in there? Okay.
So when they get in there, we'll keep our eyes open for a soft coral, Nephthia, to see if that's in there.
Sorry, Art.
So this is going to be Canister 5, right?
Mm-hmm.
If just hoover up the reef.
I know, right? That's what it feels like we did today. So everything we see, just hose it in. We'll be up till Saturday. We're sorting samples.
We're going to have a blast tonight.
We're pretty fast at that.
It's because I know what you're doing, you know, and I think you know what I'm doing.
Mm-hmm. Mm-hmm.
Ah.
Mm-hmm. We can look at the fuzzies when we're done with the.
I think that's a great
Ooh,
idea.
ooh, Medusa.
Medusa.
Squish, squish.
Turn on the suction.
Yeah, we've got some, I don't know, soft fan-like items.
Tumbleweeds?
Yeah, tumbleweeds, right? Like 11 in the sand.
And we're going to take guesses in advance of what those could be?
My guess is going to be some kind of sea pen, but they're really poofy. So I don't know.
Sea pen.
Yeah, what else would be growing out of the sediment like that? Unless it's like solidified on rock underneath that we just can't see.
I'm going to go out on a limb and say there's a hydroid there.
One of the things I haven't seen yet is Acanella, which is a bamboo coral that grows as a small in sediment.
That could be that.
Acanella, then. I think that's a good guess.
Mm-hmm.
They're really cool because they can also grow on the rock, and so they modify the holdfast, which looks like a calcareous disc when they're on the rock, into long roots when they're in the sediment.
Oh, nice.
It has all the world looking like wax. The calcareous stuff looks like wax down, and that's the form of the root.
I want to see this now.
Yeah, I think, Mary, your mic's open again.
Eurotychus got away. They don't have too far to go to find another Chrysogorgia.
Yeah, we just need one or two polyps from this. You don't need to take too much. And it's going to be pretty hard. So I have to... Yeah, try and get the living tissue, which is going to be a little pink. Probably closer to the bottom.
So we're going to dedicate this collection to my mother-in-law, Marsha Johnson, whose birthday it was yesterday.
Oh, happy birthday, Marsha.
You can say happy birthday, BB.
Happy birthday, BB.
That's her nickname. It's Grandma in Swahili.
Oh, cool. I use me mom from my grandmother.
I hope she's listening. She's a very special person. I like Meemaw. That's
yeah.
really good.
Yeah.
Stephanie.
Hey, John. What's up?
You see in the field of view. Pardon.
Go ahead. I'm listening.
Can you hear me?
Yeah.
Oh. In the field of view of camera one, there's like little clumps of. It looks like coral.
It is. It is
scattered around.
Yeah, it absolutely is.
8 o'clock, 9 o'clock.
Yeah, it's pretty much everywhere up here. It's just all dead. It's dead standing, and I think it's probably the same species we've been seeing that looks yellow. That's either Eniolapsamia or the one I can't. Solensmilia. So we're gonna grab a piece of this living and.
Okay. Have you got a good zoom of it? Make sure it's not Lopelia.
Yes, we have. Can we zoom in on this before you guys go after it? The laser's on.
I don't know what you're reflecting there.
Thank you.
Like to step over at 9 o'clock. is what I'm looking at.
Oh, okay. So that's all dead.
Like little balls. Yeah, they're dead. They're balls of dead colonial something.
Everything we've zoomed in on looks more like this pink one that's living that we're about to collect. I don't think it's Lophelia. I think it's this other species. But we can get a good zoom on the rubble over there when we're done. Let me get this last collection, and then we'll take a peek.
And I think that is a Nephthiid, just to the...
Okay.
Work? Okay. Oh, sorry. Okay, so it's right at the tip of where your hose is. In the video, I'm sure it's not like that in real life, but from what I'm seeing, right below the fluffy white thing, it's pink. It's pink, and it's a hard coral. So if you're to spin a little to the right, so a little to 3 o'clock. Yep. And then zoom. I can give you a better shot of what I'm looking at. Yeah. Mhm. Yes. So it's behind the fine coral. It's the hard orange behind the fine coral that you were just said.
Squid bombing us.
Oh, all right.
Now, Mike Vecchione was saying that if we were diving at night, these Elix squid would be all up through the water column feeding.
Neat.
And they come down here during the day. So
Yeah, and I just need a couple polyps to get a good idea. Yeah, it does. I think so. Yeah, see the orange sticking out? Yeah, little polyps at the bottom. They're going to be pretty hard.
But they will break.
You might have to smash it a little bit. Yeah, it's going to be further down. Yep. Try there. There you go.